professor in BJORKHEM. He's from the Karolinska Institute in Sweden, and he has a tremendous knowledge about bile acid synthesis. Please, speak.
Thank you very much. Thank you very much for this introduction, and also I'm very grateful to the organizers for the kind invitation to this nice meeting. My adventure with CTX started in 1978. At that time, my scientific focus was on bile acid synthesis, and I had developed a number of assays for intermediates in bile acid synthesis based on isotope dilution mass spectrometry. And I collaborated with a Norwegian group at that time, and this Norwegian group, they informed me that they had got a diagnosis of CTX in two sisters living in a very remote area in the southern part of Norway. And these two sisters had gallstones, and it was decided that they should be subjected to cholecystectomy. And in connection with that, it was possible to get permission from an ethical committee and also from the patients to get a piece of biopsy of liver from them to analyze bile acid intermediates. And we did that, and we found that the substrates for the CYP27 hydroxylase, the sterol 27 hydroxylase, were markedly increased. Some intermediates were increased more than 100-fold. And that suggested that the sterol 27 hydroxylase was deficient. And we tried to publish that, and that was very, very difficult because some years previously an American group had published a paper that suggesting that it was not 27 hydroxylase, it was the 24 beta hydroxylase that was deficient. So we had really hard time to get this paper published. But after that we had asked the editorial board that they should not consult some scientists as referees, then we got it published in JCI. This is the paper that demonstrated that CYP27 is the deficient enzyme in the synthesis. And 11 years later, we were lucky to get the confirmation of this because David Russell and his group in Dallas, they cloned the human CYP27, and they could also show that two patients with CTX had a mutation in CYP27. This enzyme catalyzes 27 hydroxylase. This is the final methyl group in the steroid side chain. And when cholesterol is attacked, the product will be 27 hydroxycholesterol. But there's not only one possibility, one hydroxylation, but also two more hydroxylations, which will give the corresponding steroid acid, cholestinoic acid. So when the enzyme is present in very high amounts, there will be a formation of cholestinoic acid instead. But the natural or the most important substrates for this hydroxylase is not cholesterol itself. This is a number of intermediates in bile acid synthesis. And when these intermediates have a 7-alpha hydroxyl group, and when that hydroxyl group is present, this is the 7-alpha hydroxyl group. When that is present, the enzyme is more active, the CYP27 is more active, and you will get 27 hydroxylated intermediates. And we believe that at these 2 points in the sequence that is the most important 27 hydroxylation occurring. So this step is obligatory in connection with formation of the bile acid quinolyoxycholic acid and cholic acid. But there is some also formation of bile acids through a pathway that starts with a 27 hydroxylation of cholesterol. Whilst all these reactions occur in the liver, this reaction can occur in almost all cells of the body. And it has been shown that in human about 10% of all bile acids are formed by this pathway by a first hydroxylation of cholesterol. Anyway, in connection with our experiments with these two patients, there was a tremendous accumulation of the substrates for the 27 hydroxylase in this pathway. For example, this SUP, the C4 compound, 7-alpha-hydroxy-4-cholesterol was increased, I think about 100-fold. So, as I said, this enzyme is present not only in the liver but in almost all cells of the body. It requires an LPH, oxygen, and oxygen reductase as electron carrier for enzymatic activity. This enzyme is located at the inner mitochondrial membranes, which means that the substrate cholesterol must be transported into the mitochondria, to this in the mitochondrial membrane, to be hydroxylated. And it has been shown that this is a limiting factor for enzymatic activity, this transport mechanism. As we have discussed before today, CTX is caused by mutations in this gene, and in some rare cases heterozygotes may have a mutation also in another gene of importance for CYP27 activity, but this gene has not yet been defined. As you know, the clinical picture involves xanthomas of the brain, skin and tendons, and you may get neurological disturbances and dementia. And biochemical, the patients have markedly reduced levels of 27 hydroxycholesterol in the circulation, which we believe is the best diagnostic marker. They have normal or low cholesterol levels, increased levels of cholesterol, the saturated analog to cholesterol, and marked increased levels of abnormal 25 hydroxylated bile alcohols. This is the general picture of patients with CTX. We just learned that Achilles xanthomas is not present in perhaps 25% of the cases. In serious cases you will also get xanthomas in the brain, in particular in cerebellum. We will hear more about the different mutations, but among the 30 different mutations that are known in the adenosine binding region or the heme ligand binding site, these mutations may be missense, nonsense, frame shift, or splice junction mutations. As I said, at present only mutations in the CYP27 gene has been defined, but there must be also some other genes that can affect the enzyme activity when mutated. This sterol 27 hydroxylase is in principle an anti-atherogenic enzyme because its role in extrahepatic cells is to some extent to remove cholesterol. 27 hydroxycholesterol, in particular cholesterolic acid, are very rapidly transported across the cell membranes to the surroundings, and that makes it possible to eliminate cholesterol by this enzyme. In particular, we could show that macrophages, and in particular pulmonary macrophages, have very, very high levels of CYP27. And we believe that the 27 hydroxyl cholesterol that we find in circulation, about 50% of it comes from the lungs. And probably 27-hydroxylase has a role in the elimination of cholesterol from the lung. Treatment of CTX with quinoxycholic acid is effective, as we already have discussed, and even xanthomas in the brain may be reduced in size. And this means that the lack of CYP27A1 mediated flux of these oxygenated metabolites, they cannot be key mechanisms behind the formation of xanthomas. There must be a continuous formation by some mechanism to explain this. The accumulation of cholesterol seems to be the driving force in connection with the development of xanthomas in CTX. So there is no development of xanthomas if not cholesterol is accumulating. Because of that, it was very interesting to try to define the origin of cholesterol. We did some experiments a long time ago where I introduced synthetically a tritium atom in 7-alpha position of cholesterol. Then I mixed this with C14 labeled cholesterol. So I had a certain ratio between tritium and C14 in this mixture. And then that mixture was injected into a rat. And then we found that the cholesterol that we could isolate from this rat had lost 15-20% of tritium. And the situation was similar when I treated myself with this mixture. There was a loss of perhaps 20% or so of the tritium. But when I injected it in a CTX patient, one of the 2 Norwegian sisters, the ratio between tritium and C14 dropped dramatically to just 20% of the original. And that means that this tritium had been lost in connection with the conversion into cholesterol. And we know that in connection with the formation of bile acids, the 7-alpha hydrogen is replaced with an hydroxyl group stereochemically, and thus it's logic that 7-alpha hydroxylated intermediate, there is a 7-alpha hydroxylated intermediate in the formation of cholesterol in patients with CTX. We had shown that one of the compounds, intermediates in bile acid synthesis, accumulating most extensively was this 7-alpha-hydroxy-4 cholestyne-threone. That was 100-fold higher levels than normal. And we could show in vitro with different preparations from different organs that this compound is dehydrated to give 3-oxo-delta-4 delta-6 compound, and that the compound was rapidly saturated to give the corresponding 3-oxo-delta-4 compound, which gave cholesterol in a high yield. So this is a reasonable mechanism by which cholesterol is formed, and we could further demonstrate that this is the most likely case in CTX. Because we found in 2 patients with CTX that while this compound was increased in the circulation 90 to 160-fold, the next intermediate in the sequence that we had found was accumulated 17 to 24 times, and this compound was accumulated 60 to 250-fold in the serum of patient with CTX, and cholesterol was accumulated 5 to 9-fold in these 2 patients. So that's it seems likely that this is the sequence of reactions that leads to cholesterol. And that means that 7-alpha-hydroxy-4-cholestyl-trione, the one that we call C4, is a crucial metabolite here. This is an intermediate in bile acid synthesis, and as you know, you have a feedback inhibition of the cholesterol 7-alpha-hydroxylase, or CYP7A1, by bile acids. We have shown that in particular ketohydroxycholic acid is the most effective suppressor in humans, in human bile acid synthesis. So this negative feedback inhibition will be destroyed if you have a block of the 27-hydroxylation. Then there will be increased activity of the 7-alpha-hydroxylase, which yields high levels of 7-alpha-hydroxycholesterol and C4. And this very high accumulation will give the result that abnormal pathways are activated. So one of them is this formation of cholesterol as a consequence of the 100-fold accumulation of C4. And also as a result of this, there will be a 25 hydroxylation of the steroid side chain, which will give 25 hydroxylated bile alcohols. And these bile alcohols can be formed in gram amounts and excreted in feces and bile of patients with CTX. It's well known that cholesterol does not pass the blood-brain barrier unless the blood-brain barrier is in some way disrupted. And cholesterol is in the same situation, that there will be no transport of cholesterol across the blood-brain barrier. But we believe that maybe C4, 7-alpha-hydroxy-4-cholestyl-trione, could pass this barrier. That was our hypothesis, at least. And we collaborated with a group in Austria who had set up a blood-brain barrier model with cultured endothelial cells from piglets, from brain of piglets. And these cells were cultured on a filter, and that functioned as a model of the blood-brain barrier. You had an apical introduction of fluid here, and then you press it through the filter and see what goes through. And it was shown that in similarity with the blood-brain barrier, there was no passage at all of labeled cholesterol. There was some passage of 7-alpha-hydroxycholesterol, and in particular 27-hydroxycholesterol, whereas the C4 was very efficiently passing here, which demonstrates that this compound can pass the blood-brain barrier. And we could also show that this compound, when the deuterium labeled and added to different cells of the brain, there was a rather efficient formation of cholesterol with 4-cholestyl-trione as an intermediate. And we could show that 4-cholestyl-trione were formed in neuroblastoma cells and astrocytoma cells and microglial cells. And also there was a production of cholesterol. So that means that there is good reason to believe that this is the mechanism by which brain xanthomas are formed, that C4 goes into the brain and then converted into cholesterol. In collaboration with an Israeli group, where we have developed and characterized the mouse model with disrupted CYP27A1. And to our disappointment, there were no xanthomas formation in these mice. They had normal levels of cholesterol and reduced bile acid synthesis as was predicted. And they had increased activity of cholesterol 7-alpha-hydroxylase, but not to the extent found in humans. The activity was increased just 5-fold or so, whereas in man it's increased more than 20-fold. That has been shown by Salin and collaborators. And because of the fact that 7-alpha-hydroxylase consumes cholesterol, there will be a compensatory increase in cholesterol synthesis, increased activity of the HMG-CoA reductase. There was slightly increased plasma levels of cholesterol, but not the marked increase that we see in humans. Slight increased levels of 25-hydroxylated bioalcohols. And all these changes were reversed by treatment with 0.05% cholic acid. Why do these mice not develop xanthomas? And we believe that this is related to the activity of CYP7A1. Cholic acid is the most effective suppressor in man of the CYP7A1, whereas cholic acid is the most effective one in mice. This may be the explanation why we do not get this high stimulation of CYP7A1 in the mice. There is also a stimulation in mouse of another enzyme, CYP3A, an enzyme that is involved in drug metabolism and has a very broad substance specificity. But it's unclear if this could be a part of the explanation. We don't believe that this is so important. In mice, in controlled mice, or knockout mice, on a controlled diet, we have a certain accumulation of cholesterol in the brain. And we subjected such a mouse to intraperitoneal injections of C4. And then we found that there was fourfold increase, 3 to 4-fold increase in the cholesterol levels in the brain, which is in agreement with our hypothesis. And we also synthesized deuterium labeled C4 and injected intraperitoneally. And then we found that after 10 days of treatment there was a significant incorporation of deuterium in cholesterol in cerebellum and cerebrum, which is also in agreement with our hypothesis. So the results are consistent with that C4 is a precursor to plasma cholesterol in these mice, but not in wild-type mice. And they may be a precursor to brain cholesterol in CYP27 knockout mice, but not in wild-type mice. And if C4 is a precursor to cholesterol, a relative high concentration is needed, more than 400 nanograms per mil, which means that you have to have an accumulation in order to get this passage. So high levels of C4 in circulation leads to transport across the blood-brain barrier and synthesis of cholesterol. And we also could show that this is reversible with cholic acid treatment. Then it's difficult to understand why this accumulation of cholesterol is accompanied by accumulation of cholesterol, because the xanthomas contain perhaps maximum of 20-30% cholesterol, and the rest is cholesterol. And we believe that the reason for this is that cholesterol is less effective as a synthesis, cholesterol synthesis inhibitor, inhibition of HMG-CoA reductase than cholesterol. And dilution of the cholesterol pool with cholesterol may thus result in increased cholesterol synthesis. We have also considered that there may be some conversion of the cholesterol into cholesterol, but we have clearly excluded that possibility now. This is some experiment performed by Magnus, and he isolated human macrophages and subjected them to different concentrations of cholesterol. And if cholesterol had been an efficient or as efficient suppressor of cholesterol synthesis as cholesterol itself, the addition of cholesterol would have resulted in reduced levels of cholesterol. But that was not the case. And that was further substantiated by looking at an intermediate in cholesterol synthesis, lathosterol, which is a marker of cholesterol synthesis. And that was not very much affected by the addition of the cholesterol. So this supports our hypothesis that the addition of cholesterol has no possibility to reduce synthesis of cholesterol, which means that cholesterol synthesis could be increased. I would like also to give a comment on a very recent paper that was very interesting. There was a mapping of different genes in connection with amyotrophic lateral sclerosis. This study identified CYP27 as a candidate gene for sporadic ALS, and that CYP27 was thus a candidate gene for this. And it was also discussed that there are some clinical mimics between ALS and CTX, and because of that it was also of interest. Recently we performed a study on 50 patients with ALS and measured 27 hydroxycholesterol in these patients. But these levels were normal, and there was no relation between the length of survival or diagnosis of ALS. So we don't believe that this is important. I would also like to give a comment on our discussion today about the best way to diagnose CTX. And we, as I said before, we believe that 27-hydroxycholesterol in combination with measurements of 7-alpha-hydroxycholesterol is a very efficient diagnostic tool. And these two compounds, 27-hydroxycholesterol and 7-alpha-hydroxycholesterol, are do we get in the same chromatogram. And this is, for example, one patient with a very serious CTX clinical picture and less than 5 nanograms per mil of 27-hydroxycholesterol. Normally it's 90 to 230. When this patient was not treated, there was very high levels of 7-alpha-hydroxycholesterol, at least 30-fold increase in this level. If we had measured C4, it would have been even more increased. And when we first treated this patient, 7-alpha-hydroxycholesterol did not go back to normal levels, but when we increased the dose of chenodeoxycholic acid, it was a normal value. And this is another patient in the same situation, where we can clearly see the treatment effect. Here we have a patient that has very mild form of CTX, and the levels of 27-hydroxycholesterol are a bit higher than in the other two cases. That also meant that 7-alpha-hydroxycholesterol was not so much increased as in the other cases. And also here we got a treatment effect. The major conclusions are that accumulation of cholesterol in patients with CTX is likely to be of critical importance for the formation of xanthomas. And this is most probably a consequence of the marked accumulation of 7-alpha-hydroxylated bile acid intermediates. And one of these intermediates, the C4, passes the blood-brain barrier efficiently and is converted into cholesterol in the brain. And it seems likely that the accumulation of cholesterol in the brain xanthomas is secondary to the accumulation of cholesterol. These are my collaborators in these studies. Thank you very much for your attention.
Thank you very much, Professor BJORKHEM, for this excellent lecture. I think there's plenty of room for discussion later on this afternoon. And, I would like to introduce, but introduction is not necessary anymore, I think, Maria Jesus Sobrido from the New Genetics Research Group, Santiago de Compostela. She's going to talk about genetic analysis and the CTX genetic database.